my dear little champions, I welcome you all to PU2 Brahmastra Series. Today is day 6 and today we are going to start off the chapter that is molecular basis of inheritance, and I am telling you, if you are going to be in this lecture with me throughout, then I am guaranteeing you to give you 13 marks because this chapter carries 13 marks, and which is why it is very much important. We cannot ignore it. And what type of questions can you expect coming from this particular chapter according to the blueprint? So there will be one marker question, there will be one 2 marker question, there will be 2 5 marker question, which is a big deal. So if you are scoring these 2 5 marker questions, my dear children, 10 marks so anyway you will get, but we are looking for 13 and nothing less, right? And what type of questions and how many of them will I be solving in this session? 10 1 marker question, 7 2 marker question, and 11 5 marker question. So I'll make sure that your concepts are also getting cleared accordingly with the questions also which I am solving. Okay, wherever you need a concept support, I will be providing it to you. That's my promise. And what should be your promise? Your promise is to stay active throughout this session, listen to me very carefully and try to solve the questions by yourself also. Okay, that should be the deal. So let's get started with the first question that we are having over here. The number of nucleotide pairs present in the DNA of the primary oocyte of a newborn in human is. What are we trying to find, children? Number of nucleotide pairs. Where, children? In the primary oocyte. Now if you know, primary oocyte is a stage in the gamete formation of the female. So I'll suppose this is gamete only that I'm talking about. And gametes. Gametes are different from all the normal cells of our body. Why? Because gametes will be having half the number of chromosomes. Half the number of chromosomes means half the number of DNA as well. So basically, I will say that they are haploid. Why are they haploid? My dear children, one sperm is there having half the number of chromosome, one egg is there having half the number of chromosome, comes together, forms the zygote having the normal number of chromosomes. So haploid comes, forms something that is known as diploid, that has double set of chromosome. That is totally fine, the actual set of chromosome that we actually needed. Okay, so gametes have half the number of chromosome. So if I say a full cell, a normal cell has 6.6 into 10 to the power 9, what say base pairs of DNA and things like that. Basically, what will be the half of it? It will be 3.3 into 10 to the power 9. So this becomes the answer for your model question which came in that has been put up in this particular question paper, 2024-25 question paper. Works? I hope you got it. Next thing. Pyrimidine base present in RNA in place of thymine of DNA is. What do you mean by pyrimidines? What do you mean by purines? Basically, all of them are nitrogenous bases which forms a major part of your DNA, my dear children. So I'll say there is 2 team. One is team purine, another one is team pyrimidine, my dear children. Works? Okay. In team purine, you have 2 contestants, A and G. A for adenine and G for guanine. Now over here in team B, which is pyrimidine, there is T and there is C. T for thymine and C for cytosine. Now I'll tell you one more thing. Okay, A is best friends with T and G is bestest friends with C. So A and T has 2 bonds which are 2 hydrogen bonds. G and C have 3 hydrogen bonds over here. So this is the case of a normal DNA. But in the case of RNA, things are going to change. So in the case of RNA, instead of T, A will be binding with some other pyrimidine which is known as uracil, my dear children. So RNA does not have T, hence A will be binding with U. G will still be binding with C. This is the bestest bond that they have, right? So RNA has uracil, please remember that. So this is the pyrimidine. So pyrimidine base present in the RNA, that is in place of the thymine of DNA is what? Uracil, my dearest little champions. So this becomes your answer. Cool? Okay. Now getting into the next question. In lac operon, if mutation occurs in the Z gene, okay, in the lac operon, if mutation is occurring in the Z gene, so there are 3 genes, Z, Y, A. Z, you can remember like that. Okay, Z1, if it is mutated, what will not be produced over here? That is the question. Trans-acetylase will not be synthesized, beta galactosidase will not be synthesized, permease will not be synthesized, lactose digestion will be rapid. So basically Z gene It's a gene, so it has to code for a protein. And what protein is it coding for, children? Z gene is coding for something that is known as beta galactosidase. Okay, now Y gene is coding for something that is known as another protein that is known as permease. A gene will be coding for another protein that is known as transacetylase. So basically, why is ma'am saying protein, protein, protein? Aren't these enzymes? Bachcha log, enzymes that you see over here, they're all proteins only. So Z gene is coding for this, Y is coding for this, A is coding for what? Transacetylase. So if I am getting this mutated over here, there is some mutations happening over here. What will not be produced? Beta galactosidase would not be produced. So what will be the answer, my dear children? It will be the answer. This came again in the model question paper 2024-25. Works? Let's go on to the next question. Yes. A DNA segment has a total of 1000 nucleotides. How many, children? 1000 nucleotides, out of which 240 of them are adenine. So we know adenine is 240. Cool. How many pyrimidine bases this DNA segment possesses? How many pyrimidine bases are there? That means they are asking you a total of T as well as C. How can you find a total of T as well as C? My dear children, if adenine is 240, adenine always pairs with thymine. So thymine will also be what? 240. That we know. So thymine is basically 240. Done. Now we know 240 plus 240 will be is equals to what, children? 480. So 480 base pairs are already done out of 1000. Out of 1000, 480 base pairs, we are already spoke about that. So let's minus. Let's minus the next thing, and then we will be able to find the number of what? G and C together. If you minus this particular amount with 1000, we would be able to find the total number of this thing. So 1000 minus 480, you will be getting, I think it will be 520. It will be what, children? 520. Now 520 is the total of G and C. If you want to halve 520, if you want to halve 520, then what you get? 260. Agree? So can I say this is 260? If this is 260, this will also be what, children? 260. Now what you have to find out? You have to find the total number of pyrimidines. So T plus C. So 240 plus 260, what will be the answer? 500. If you have not got, listen to me carefully. They have mentioned that adenine is 240. If adenine is 240, then thymine has to be 240 because A and T forms the pair. Right? Now, if you have the total number of these nucleotides, you can minus it from 1000 because we know total number of nucleotide is 1000. So when we minus it, we get the total number of G and C. We have not got the separate version of G and C. Okay, we have got a total number of it. Now the total number of it was 520. Now when you divide it with 2, basically what do you get, children? 260, 260. But the question is, they are asking for what, children? They are asking for pyrimidine bases, and T and C are the pyrimidine bases. So you have to plus this 240 plus 260, you get what? 500. So this becomes the answer. This is a model question that has come in this particular year's paper. Okay, so I hope you understood this thing. Works? If you have not understood, pause the video, try to look at this, you will get it. Fine? Okay, next thing. Nitrogenous base sequence in a strand of DNA is ATGCTTGA. The sequence in the complementary strand will be. This is an interesting and a very easy question. I wish you get these type of questions. So ATGCTTGA is a strand that we are having, and we have to find the complementary strand. So we have to find, what does A bind with, children? A binds with thymine. They are friends, right? So T will bind with adenine, G with C, C with G, T with, tell me, A, T with again A, G with C, A with what? T. So you have to find this over here. So TAC, okay, GAACT, GAACT. Am I seeing two options which are looking similar? No. So this is TCC. So I got this option as the correct answer. TACGAACT. If you are not aware about A binding with T and G binding with C, it becomes a problem. So I want you to remember it now only. How you can remember? There's a song AGOG, right? So AG comes under what? Purines. Okay, and T and C, You want TC from your school after completing your this thing, then you have to study as of now. So TC comes under pyrimidines. Okay, done? Cool. So this question is also over. Let's move on to the next question. All the terminator codons begin with the nucleotide of. What do you mean by terminator codon? So basically there is a process which is known as translation, where what is happening, where there is a strand of RNA which is known as messenger RNA, which gives information to form what? Proteins. Which give information to form proteins. And now this mRNA will be having certain codes which gives the information to form proteins. For example, I have A, U, G, then C, T, C, whatever. I have so many codons over here. These three things that you see over here, they are known as codons. So each codon is going to code for an amino acid. And when these amino acids, they come together, what are they going to make? Amino acid plus amino acid plus amino acid plus so many amino acids gives you what? Proteins, which is why I say that this mRNA is going to code for proteins. Now, this process has to end somewhere or halt somewhere. This process is known as translation, and it has to stop somewhere. It will only stop when it sees a different type of codon, which could be UAA, which could be UGA or UAG. So you see these three codons. If you find these three codons over here, that means the process of translation will be stopping over there. So UAA, UGA, UAG, UA, UGA, UAG. Okay, they all are known as terminator codons. Now, all the terminator codons begin with the nucleotide of what? If you see all of them, they are beginning with what? Uracil, my dear champions. That does not mean others which are being made and starting from U are stop codons. No, only these three other stop codons, you have to remember it. UAA, UGA, UAG. Okay, so this will be uracil. Works. I'll also tell you one more thing. AUG that you see over here, this is a start codon. So every mRNA should be having this for starting off this process. So AUG is a start codon. These are the stop codons. Works? Chalo, next question. The semi-conservative mode of DNA replication was demonstrated by. What do you mean by semi-conservative mode of DNA replication? As in, for example, suppose this is a DNA and this DNA has to make a copy of it. So what happens is that they will unwind. Okay, there is one strand over here, there is another strand over here. Now, these strands will be known as what? Parental strand, because I am getting it from the parent. Now, there will be a new strand being made up right over here. And now, this is the new DNA that I am having over here. So, why do I call this semi-conservative? Because I am conserving one parental strand and I am making a new strand over here. So, semi as in half of it is being conserved and half of this is new. So, this particular thing, semi-conservative mode of DNA replication, was demonstrated by none other than Michelson and Stahl. Okay, it's a direct question. Michelson and Stahl were the one behind it. Okay, works? Cool. Next, what is the name of the process where the introns are removed and the exons are joined together in defined order? What do you mean by introns? What do you mean by exons? Exons are the one that will be actually coding for proteins. You saw, right, in RNA and all, there are certain strands which is coding for proteins, right? So there will be certain area which will be coding for protein, and then comes certain area which does not code for anything, it's just there. What do you call that particular region as? You call them as intron. So you have, suppose, this is intron, this is intron. Now over here, I just want, this is intron and this is also intron. What about the other areas marked in yellow? They are exons. Exons are the one that will express and they will form proteins. Okay, they will form proteins. So I do not want these introns. So I have to what? Cut them off from here. And then I'll get a strand like this which just contains the exon. What is this particular process known as? It is known as splicing. What do you call this process as? Splicing, where I'm cutting it off. Okay, what am I cutting? Introns, which are not coding anything. I don't want them. I just want exons. Fine? Okay, one very important clue. Introns are found in eukaryotes, not in prokaryotes. Okay, so just remember that. I just thought of it, so I thought of telling it to you also. Now, question number 9. What is the process of activation of amino acids in the presence of ATP and its linkage to their cognate tRNA known as? What do you mean by that, ma'am? So basically, I told you there is an mRNA. mRNA has codons like AUG and things like that. And I told you that amino acids, basically each codon is coding for an amino acid, right? So who brings this amino acid? There is a very important type of tRNA which you call as, very important type of RNA which you call as tRNA, my dear children. So tRNA, which is a normal RNA, what happens is that on the 3' end of it, amino acid will be coming and binding. And this amino acid specific. How is it specific? Well, here you have AUG. Over here in the tRNA, you will be having the opposite of it. If A is here, this is RNA, not DNA. So if A is here, what will be over here, children? U or T? It will be U. Over here U is there, so it will be A. Over here J is there, so it will be C. So this guy, no, with the tRNA, it comes like finding, oh, I see AUG, let me go and bind over there. Well, this particular tRNA has an amino acid attached to it, and this process of addition of this particular amino acid to this particular tRNA is known as charging of tRNA, or you call it as amino acylation of tRNA. What do you call it? Charging of tRNA, or what do you call it? You call it amino, listen to me very carefully, okay? Amino acylation of tRNA. So over here, charging of tRNA is there, so I'll say this is correct. But let me read other options. Charging of ATP, no. Amino acetylation tRNA, amino acetylation of ATP, what are we talking about? Charging of tRNA or amino acylation of tRNA. So charging of tRNA is correct, not acetylation, it's acylation. Works. And here you require ATP. Here you require what? ATP, my dearest champions. I hope you got that also. Okay. Now last question from the MCQ series. The first genetic material could be what? Protein, carbohydrate, DNA or RNA. What could be the first genetic material? So in your textbook there is something like RNA world. So according to RNA world, RNA is the first genetic material. Okay, but RNA had its own drawback, that is it was not that stable. Okay, it is that extrovert friend in your gang who talks to everyone. So basically this particular RNA is something that can go react with lot of things. Okay, so that was becoming a problem, because if it goes and react, it will undergo certain changes in itself, and suppose if it happens, then you are basically changing up the whole structure of an organism. We do not want that. So we went from RNA to having what? DNA, which is a more stable form. It's not like we made it, nature itself preferred it, and that's how it was being made. So RNA was the first genetic material, my dear children. This came in March 2023. Okay, works. Now let's get started with the 2-marker question, and let me just tie my hair. Can you give me a gap for that? And let's read it. Which sequence of bases transcribed from DNA are found both in hnRNA and mRNA? What do you mean by hnRNA? And what do you mean by mRNA? I'll tell you. Basically, the question says, what is the sequence that is common between something that is known as hnRNA and mRNA? So, hnRNA stands for heterogeneous nuclear RNA. What do you call it? Heterogeneous nuclear RNA. And mRNA stands for messenger RNA. Now I want to give you a background check. Basically, you know this is a cell. Okay, inside the cell you have the nucleus. Inside the nucleus you have a very important thing which is known as what? DNA, which stores all the information in the form of genes. Right. Now these genes are going to code for proteins. But the problem is that for making proteins you have to take the help of one very important organelle which is present outside the nucleus. And what is this organelle known as, children? It has 2 subunits. What do you call this organelle as? Yes, it is known as ribosome. So basically, the DNA has to go out to make the proteins, but DNA cannot go out. No, it will be really risky. There are so many enzymes in there. So what do you make? We make a duplicate of it somehow. Okay, we basically make something which has all the information in the DNA and we transport it outside. So what is that something? What is that something which is being made over here? And what do you call this process? You call this process as transcription. you call this process as transcription, where with the help of the information stored in the DNA, you are going to make up a RNA. You are going to make up what, children? An RNA. Now, this RNA which is being made, okay, what do you call this process? Transcription. Okay, and this RNA which you are making, you usually say mRNA, that is messenger RNA, but no, mRNA has still not been made. It is known as hnRNA. It is known as what? hnRNA. Fine. Now, how is hnRNA different from mRNA, ma'am? Basically, I'll tell you one more thing. It's the mRNA that will be going out and meeting the ribosome for making the proteins. So how did this conversion happen? What is the change that is happening over here? So basically, hnRNA consists of both exons as well as introns. Exons and introns. Now, you know that exons are the only one that will be helping in making the proteins. So why will I need the introns over here? We have to cut this introns. And this process is known as what? We studied up B. We just right now. What do you call it? You call it splicing. So basically, hnRNA has introns and exons, whereas when you talk about the spliced version of this, it contains just the exons. And what do you call this whole structure as? Now you call it as an mRNA or messenger RNA. Now this guy transforms into messenger RNA and the messenger RNA goes out. So what is the sequence? Sequence of bases transcribed from DNA are found both in hnRNA and mRNA. It will be the exons which I will find both in hnRNA as well as mRNA. This is a 2-marker question, came in model question paper. Okay, this is how you have to write it. The sequence of bases transcribed from DNA that are found both in hnRNA and mRNA are exons. Exons are the coding regions that remain in the mRNA after splicing, while introns are removed during processing. So these were the introns, you removed it, and now you just have the exons. Works? Cool? Okay. Mention the levels where gene expression can be regulated in eukaryotes. Gene expression as in gene's sole duty is to make what? A gene's sole duty is to make a protein. Right. So in how many ways can I stop this or can I regulate this? I want to increase the expression, I want to stop the expression. What all can I do? First thing, I can mess up with the transcription that is happening over here. That is DNA, when it helps in the formation of RNA, this process is known as what? Transcription. So I can bring about changes in there, right? This is one level of things. Second thing, when the processing is happening, I can bring about changes over here. I don't want the processing to happen. If I don't want the processing to happen, obviously it won't be going out and making proteins also. You get it? So this is how I can regulate it. Okay. Apart from that, what all do we have? So transcriptional level is formation of primary transcript. There I can bring out certain changes. Second thing, processing level. Over here, I can regulate the splicing and everything. Then also it will be stopped, or basically whatever has to happen could be happening. Then transport of mRNA from the nucleus to the cytoplasm. So mRNA is over here in the nucleus. You have to take it out. Right. So over here also I can do something. I am the villain over here. I can do something such that the mRNA does not get transported outside. Now if mRNA is not going outside, will it make the proteins, my dear children? No. Okay. And suppose mRNA has got outside also. It met the ribosome. But now I am not allowing the next process to take place, that is translation. Remember the tRNA coming with the amino acid? If I stop that process in whole, don't you think that I won't be able to make the proteins? Yes, I won't be able to make the proteins. So these are the levels in which basically the gene expression can be regulated in the case of eukaryotes. Eukaryotes are organisms that has a nucleus, that have membrane bound organelles. Okay, so what are the levels? Transcriptional level, okay, where I am making the hnRNA thing. Second, processing level, okay, where the hnRNA is bringing down changes to form a mRNA. Then transport of mRNA level, at that point of time I can bring out certain changes. Then translational level. Will you be able to remember it now? I hope I am explaining you the concept. I might be going a bit fast. Why? Because this lecture might be too long and you might get bored also. So I am using my 100% energy to teach you, my dear children. So I hope you are vibing with me. We are studying together, right? And I hope I am making you understand the concepts as well. So will you remember this? These are the levels. Please remember, this came in model question paper. Okay, this year's model question paper. Going ahead, write a note on the role of sigma factor and rho factor in transcription in prokaryotes. So what is sigma factor and what is rho factor in the transcription process? So what do you mean by transcription? I again told you, transcription is basically when you have the DNA and now you want to form an RNA out of it. Usually we say it an mRNA or messenger RNA. So basically this process is not as easy as you see. Okay, so basically first thing, there is 3' to 5' strand of DNA and the complementary strand. DNA is coiled, right? So if I am opening it, one is 3' to 5' strand, other one will be 5' to 3' strand. Okay, now over here there is a region which is known as promoter region. What is this region? Promoter region. Now over here at the end there is another region which is known as terminator region, and this area is where the transcription will be actually happening. Okay, this is the gene that we want to transcribe. Okay, so over here promoter region is there, transcription region is there, and there should be one very important enzyme for transcription, and that enzyme's name is what? RNA polymerase, my dear children. So RNA polymerase is the one that will be making an RNA out of it. For example, this is 3' to 5' and RNA polymerase always works in 5 to 3 direction. Okay, RNA polymerase always work in 5 to 3 direction. So it won't go and bind over here. That is not what I mean. It will find the opposite strand, that is 3' to 5, and now from here it will start to what? Transcribe things. So suppose here there is A in DNA. In the case of RNA, here it will be U. Here if it is T in the case of DNA, here it will be A. Here it is G, then it will be C. Just like that, you are forming what? An RNA. In which direction? From 5' to 3' direction. But it's not as easy as it seems. When this RNA polymerase comes in the promoter region, it has to come with one more guy which is known as the sigma factor. Without sigma factor, it cannot bind to the promoter region and begin this particular process. One sigma factor is over here. Once you have sigma factor with your RNA polymerase, that's when it will come and bind on the promoter region, and that's when it will initiate the process of transcription. Similarly, then this guy falls off. Then the sigma factor goes off. After initiating, it just goes off. Then later on, when the RNA polymerase reaches the terminator region, at this point of time, if you want to terminate, end the process of transcription, you need the support of another guy, which is what? Rho factor. Which is what? Rho factor. So RNA polymerase needs two friends in the whole process. One is sigma factor and the other one is what? Rho factor. The rho factor comes and attaches, that's when the termination actually occurs. You get it? So sigma in the promoter region and rho in the terminator region. And this is how you have to write the answer. Sigma factor binds to RNA polymerase, guiding it to the promoter region for transcription initiation. It detaches after the initiation. As you see over here, this is sigma factor. If you are not able to see, I will zoom in. Zoom, 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 zoom. Yeah. This is sigma factor that has come and attached. Well, you see transcription have already started. This is a new strand of RNA that is being formed. At this point of time, these guys dissociate. The sigma factor say tata bye-bye and I will go. Okay. Over here, when you want detachment of RNA polymerase. So at that point of time, what do you need? Rho factor. Okay. So in termination, you need rho factor. I hope I was able to explain you this as well. Works? Children, you are enjoying this session, or basically you are getting bored, you are getting stressed? What is the case? I hope you are enjoying, and I want to remind you children that you would be able to do it. Do not worry, everything will fall in its own place. You just stay calm and you give your best. That matters. Okay? Cool? Chalo. Question number 14. What are VNTRs? Mention the steps to detect VNTRs in identifying criminal in forensic investigations. So CBI, come over here. Let's see. Those of you who are interested in detective things, it will be really good for you. So VNTR means variable number of tandem repeats. What do you say? Variable number of tandem repeats. Now this question is really important because this is the same principle we are going to use for DNA fingerprinting as well, which is there in your book, and questions usually come in 5-marker session also. Okay. So variable number of tandem repeats, basically we all have a certain sequence of DNA which is very unique to us. For example, I have the sequence ACGT. This is the sequence that I am having, and I will be having so many of them in my body. In my DNA, there will be so many ACGT. Fine. That is unique to me. You might have some other sequence which might be in so much repetition. For example, you might have GC80. That might be your sequence. but it is unique to you. Okay, so number of this particular thing is unique to me, and I might have so many repeats of it. So this is what you mean by variable number of tandem repeats. Okay, so you might have bigger sequences, but they are repeated often. But what is the use of VNTRs? What are the use of these sequences that you see over here? Suppose I have committed a crime. Okay, I have committed a crime, and in the crime scene my hair follicle is there. Now the people came, they checked the hair follicle, and basically what they did was they isolated the DNA from that cell. In the follicle you get the DNA also. So you extracted that particular DNA. So this is the first step. What do I do, children? Basically I isolate the DNA. Cool? From the hair follicle of the suspect. Now doctors have a doubt that this is mine. So what can be done? Similarly, from my body also, my blood or my hair follicle again can be taken. Okay, and then they will be checking for the repeat that I am having. Suppose, okay, suppose, or probably, I don't want to complicate you. Listen to me very carefully. Okay, there's a crime scene happening. There's a crime scene happening over here, and at this particular point I got some hair. Okay, and this hair follicle was checked and found that this is the sequence that this particular person's body has, that is ACGT. Then basically there's a suspect, and the suspect is me. Okay, my DNA is also being checked. So for my DNA is taken isolated. Okay, and then what is done, children? After isolation, my DNA is being digested with the help of restriction endonucleases. So my DNA is being cut at different sites with the help of what? Restriction endonucleases. Okay, and this restriction endonucleases are made based on this particular sequence only. So basically they will be going and cutting only at this particular area where these guys are there. Only at areas where these guys are there. Okay, so this was found in the hair follicle in the crime scene, and I am a totally different person. My DNA is isolated, and my DNA is being cut using restriction endonucleases, which are very specific to these sequences. Okay, now when it is cut, it is fine. Now after it is being cut, after the DNA is being cut, no, it is run through gel electrophoresis, so that according to the length it can be separated. For example, this particular sequence was over here and here, so it was cut at this particular length. Again there was another sequence over here, the sequence was there, over here the sequence was there, so this is cut in a shorter length. So what do? You separate them out using what? Using gel electrophoresis. That is the next step. After gel electrophoresis, I am not able to view the different strands of DNA that is being formed. So what can I do is that basically I can transfer it to something else, which is what? A nitrocellulose membrane, and this is known as southern blotting, or basically you call it blotting. What do I do, children? The DNA which is over here, I am going to transfer it into a sheet, nitrocellulose sheet, which is known as southern blotting. This process is known as southern blotting. Now I have the sheet where the DNA strands are like this. Now what do I do is, I have the sequence, okay, I make a probe out of it. What do I make? A probe out of it, and I tag this probe with a radioactive label. Radioactive label as in something, when the UV rays fall on it, it will be showing up the color. Okay, so basically I am taking the probe, and probe is nothing but the sequence that I am having, and I am attaching it with what? A radioactive label. Now I am taking this probe and putting it to react with this particular thing. Now suppose if I do not have any sequence matching to the probe won't be binding, and hence when I put UV radiation, it won't be appearing as well. But when the probe was actually put, it was showing up a color change over here. That means I also have the same sequence over here. I also have what? The same sequence over here. So detection of hybridization DNA fragments by autoradiography. Okay, so what did I do? I'll tell you again, listen to me very carefully. This is important for DNA fingerprinting. Okay, first thing, there is a crime scene, there is a hair follicle. In the hair follicle, I checked for the DNA, and I found that this is a sequence which is being repeated. So this is the repeat that I am having, variable number of VNTR sequence that I am having over here. Now I have two suspects. I have two suspects over here. This is suspect number 1, this is suspect number 2. Okay, I took their DNA also, and then I isolated their DNA. I cut it with restriction endonucleases. Restriction endonucleases are enzyme that will cut the DNA from between, okay, from inside. So they cut the DNA. Okay, After cutting the DNA, I want to arrange in the form of fragments so that higher and the lower things are separated. Eventually, I can see things clearly. So that is also being done. Gel electrophoresis is done so that the fragments, they separate out. Both will be having different type of fragments and both will be separating. Then what did I do, children? I took the probe that I was having. Actually, the probe is not made out of this. The complementary stand is taken for the probe. For example, this is A, then here it will be T. Here it is C, then G, here it will be this. This will be the probe. The complementary stand will be the probe, and I will be attaching with the radioactive label. Now, this particular probe, I am going to put it on the nitrocellulose sheet. So, where the gel electrophoresis thing happened, no, I am transferring all the content to one nitrocellulose sheet, and then I am making it to act with the probe. So, the probe, when it sees the complementary base pair, it will go and bind over there. If the complementary base pair is not there, it won't be binding. When it binded, I got to understand that suspect 1 is the actual person who did the crime, because this probe is kind of unique to me. Unique to the person who is doing the crime, right? So, those people would be only having this particular thing. So, with this, I was able to understand that this is the person who did the crime. I don't know how far you understood, but I tried my level best, okay? But I think you understood a bit, right? So, isolation of DNA is the first step. Next is digestion of DNA by what? An enzyme. Then is separation of DNA fragments by what? Electrophoresis. After that, I would be transferring them. I cannot directly put the probe in the gel electrophoresis chamber. I cannot do that, which is why I need to transfer it into what? A nitrocellulose or nylon sheet. Then I will be hybridizing as in making it a complementary pair with the labeled VNTR probe. Okay, this is radio labeled VNTR probe. Now, detection of hybridization DNA fragments by auto radio. When I put UV radiation, they will be lightening up because they have found their complementary pair. Okay, got it? So, this is VNTR. This is how you have to answer. This is the same thing that you have to answer for a 5 marker. So, this is not the answer just for a 2 marker. I extended the answer. Okay, I extended the answer for a 5 marker. So, you can explain just like this for a 5 marker as well. Cool? This came in July 2018. Next question. Write the difference between euchromatin and heterochromatin. Name the virus used in Hershey and Chase's experiment. This came in July 2022. So, difference between euchromatin and heterochromatin. First thing that I want you to know is basically our DNA is kind of very long. Okay, about 2 meters long. So, 2 meter long DNA have to be put inside the nucleus of very small cell. For that, it has to be really coiled. For that, what do we have, children? We have something that are known as histone proteins. Okay, what do we have, children? Histone proteins. So, there is an octamer of histone. That means 8 histone proteins are coming together. 1, 2, 3, 4, 5, 6, 7 and 8 are there. Okay, what is the use of this histone protein? Basically, our DNA will be coiling around this histone protein almost twice. Almost twice they are coiling. Why? Because we want to allocate them into smaller space. Again, there will be the next set of histone proteins over here. Again, our DNA will be coiling around it as well. How many times, children? Twice. Got it? And there will be a histone protein over here also. This histone protein is known as H1 histone protein. This is known as what? H1 histone protein. How many times? Twice the DNA is rotating. And how many base pairs are there over here? 200 base pairs of DNA is there. And what do you call this whole structure? Where you have the histone proteins plus you have the DNA. What do you call this structure as? You call this structure as a nucleosome. You call this as a nucleosome. Now, why did I explain all about this? Because basically, when the DNA is in the loose state, okay, it is not very much coiled. You can see them separately. Okay, you can see this nucleosomes separately. And now this loose structure that we are having over here, what do you call it? Euchromatin. One more very important thing, the DNA plus the nucleosome together, you call it as a chromatin fiber. When the chromatin basically condenses together very close, when all the nucleosomes, they come very close to each other, you now call this as what? Chromosome. So, chromatin is a less condensed form of what? Of chromosome. Okay, so this chromatin fiber is over here. Now I say they are loosely arranged. So I will call this structure as what? Euchromatin. what will I call it? Euchromatin. And euchromatin, I'll say it's actively transcription is happening, everything can happen over here. So they have space for transcription and everything to happen, for enzymes to bind and things to happen. And then there is another version of chromatin that is known as heterochromatin, that is found in chromosomes. Basically what happens over there, in heterochromatin they are kind of together, all the nucleosomes, they come very, very close. This H1 protein will be very close and in between the two nucleosomes. That is what you call as heterochromatin, my dear children. So over here this is the answer. In a typical nucleus, some loosely coiled regions of chromatin are there with light stained, and transcriptionally active is known as euchromatin. So they will be taking light stain over there, okay, and they will be transcriptionally active. That means transcription can take place over there. On the other hand, the chromatin that is more densely packed and basically stains dark, it will be darker in color because they are more close, okay, and transcriptionally they are inactive. They are not, because you are not giving space for enzymes to come and do transcription, so you cannot expect that. So that is why it is known as heterochromatin. Now this is a very important question. I saw so much times it have got repeated. Okay, now the second part of the question says, name the virus used in Hershey and Chase experiment. This is a direct answer. The answer is what? Bacteriophage. Bacteriophage is a virus, it's not a bacteria. Bacteriophage is going to affect the bacteria, which is why it is known as bacteriophage. Okay, we'll talk about Hershey and Chase experiment. There is a question for that in 5 marker. Don't worry about that. Works? Next thing. List the goals of Human Genome Project. This is something that I added, seeing the time of repetition that has come. Okay, what are the goals of Human Genome Project? Now this is something that you might have to take it in your heart by heartly, but yes, I will try definitely from my end. Okay, first goal was to identify all the genes. It was thought that there are almost 20,000 to 25,000 genes over there, but apparently it was wrong. Okay, so identify all the genes in the human genome, approximately how many children? 20,000 to 25,000, that was the first goal. Second goal, determine the complete sequence of DNA. Apart from finding the genes, the DNA also, this complete sequence of the DNA of what? The human genome was something to be found according to the goals of Human Genome Project. Okay, next thing, once you have found all this thing, you have to store this information where, children? In the database. Okay, now do what? Once you have stored information in the database, you have to improve the tools. For what? Analysis. You have stored it, but you are not able to find it. That's a problem. So that is the next goal, to improve the tools. Next thing, you have improved the tools, everything is nice. What you can do is give this information to other areas of development also, so that they can also do their research. So transfer related information to other sectors and industries. Next thing, and the most important thing, address ethical, legal and social issues, that is ELC, arising from genome studies. You have to address it. It's not like, for example, I am going to kill a monkey for finding the genome. I cannot do that always. That is not at all ethical. So these things have to be addressed and you have to find a solution for that as well. So what are the goals, children? First thing, to identify all the genes. Almost how many? 20 to 25. Then I have to find all the sequence of the DNA also. Determine the sequence of all the complete DNA that I have. Next thing, I have to store this information where? In the database. Next thing, I have to improve the tools also, because I want to find the data, and that should be possible. Next thing, to transfer this information to other areas which are doing research. Next thing, address the ethical, legal and the social issues that could come from this, that is LC. Okay, got it? So I hope I didn't help you mug things. I hope I helped you to understand this particular answer. So these are the goals of Human Genome Project. Now there will be salient features of Human Genome Project that will be asked for 5 marker. I will be discussing about that as well. Okay. Now mention the criteria for a molecule that acts as a genetic material. So for example, I have RNA, I have DNA. I say I want DNA to be the genetic material instead of RNA. Why did I choose that? I told you that RNA is more reactive. RNA does not have that particular feature to be the genetic material. Genetic material has to be safe, has to be stable. So what are the other criteria that a molecule should be having to be acting like a genetic material? I think this is the last question from a 2 marker. Okay, let's see. So it should be able to generate its replica. There should be a replica copy that could be made from this. For DNA, it undergoes replication, so it is okay. Because obviously you have to pass this to next generation, to all the cells also, right? For example, cell divides and does not get the DNA, does not work, right? So we want replication to happen. So it should generate its replica. Next thing, it should be chemically and structurally stable. It should not go around reacting with a lot of things. Okay, it should be stable. It should provide the scope for slow changes. It's not like I don't want any bit of changes in the DNA. I definitely want some changes that should be good for me. So this is known as these changes that happens, what do you call them? Mutations. So mutations can be good, mutations can be bad. Bad mutation can lead to diseases. Good mutations can help us in survival. For example, there's one bacteria, there's another bacteria. One has mutation, other one does not have. This mutation is allowing the bacteria to survive in hotter climate. That means that's a good variation that it is having. So obviously, there should be a scope for what slow changes which is required for evolution. Otherwise, we would not have been evolved. We would not have been humans. Evolution has happened. Some changes happened in the DNA, which is why we are there. Hope you get it. Okay. So next thing, it should be able to express itself in the form of Mendelian character. It should be able to express in the form of Mendelian character, as in there should be certain characters that would be dominating, other one will be recessive. Okay, there should be pairs of character. Okay, there should be differences in the character. It should not be just one character. Okay, so basically a DNA should be having genes, and this genes should be coding for different character, which makes us totally different as well. Okay, so it should be able to generate the replica, should be stable, should provide a scope for what? Slow changes, and it should be also able to express the Mendelian character. So these are the criteria that we have. Okay, finishing up the 2-mark question with this, and this is a gentle reminder to have a glass of water. Okay, you can stop the video, go have a glass of water and come back. Okay, so let's begin the battle, the 5-marker questions. Okay, so list out the salient features of double helix model of DNA. How does the DNA look? For that, I want to tell you that DNA, the term stands for what? Deoxyribose. Okay, not deoxyribose, ribonucleic acid. Deoxyribonucleic acid. That is DNA. So DNA is made up of a sugar that is known as deoxyribose, which looks like this. Ribose sugar has 5 carbon. Okay, 1, 2, 3, 4, and the 5th one is over here. This is the 5th carbon. Okay, on the first carbon that you see over here, it is attached to a nitrogenous base, that is adenine, guanine, cytosine, thymine, all of them. The second one just has hydrogen. Okay, the third one, I'll say it has OH group. Third one has OH group. Now over here, the 5th one is the one that is attached to phosphate. It is attached to phosphate group. Fine. This is the structure of a nucleotide. So nucleotides are the ones that makes up, nucleotide are the one that makes up the DNA. So nucleotides come like this. Again, there will be nucleotide, and goes off again and again and again and again. Basically, how are they connected? These nucleotides, how are they connected? Nucleotides are connected with the help of this bond that I am going to show you over here. See it clearly. So the phosphate which is over here, it is attached to the next nucleotides, third carbon right over here. Since over here also you have carbon, over here also you have carbon, over here you have phosphate, and there is OH and a lot of things being released. I call this bond, you call this bond as ester bond. And since phosphate is there, I call it phosphoester bond. And now you have 2 of them, 2 such kind over here. So what do I call it? I call it phosphodiester bond. So 2 nucleotides are linked together with the help of what? Phosphodiester bond. Again there will be carbon, 5th carbon, and the 5th carbon will be having what? Phosphate group, my dear children. So this is how the things are being arranged over there. Okay, on this particular side. Now on that particular side, you need, this is one strand. This is one strand of DNA. And what is the polarity? What is the polarity? Basically over here you saw 5' end is there. 5' end has what? Phosphate group. Is it attaching to something else? No. So it is a free end. So I call it 5' end. What is it, children? 5' end. Similarly over here, you see 3 and there is OH. It's free. It is nothing, it's attached over here. So this is the 3' end which has OH group attached over here. Now what about the opposite the next complementary DNA, which strand will be here, it will look something like this. You have to make it the opposite way. So this will look something like, over here this is the first carbon, second carbon, third carbon, fourth carbon, and this will be the fifth carbon. First carbon has what? Nitrogenous base. So here I will be having the opposite nitrogen base. If here I had A, here I will be having T. Okay. Now on the third carbon, what will I have? I have, children, OH group. On the fifth carbon, what will I have, children? Phosphate group. Similarly, this will go pair up with the next nucleotide like this. So over here, if I see, I have a 3OH group that is free. At the top also I would have something. Anyways, the 3OH group will be free over here. On this side, I will be having the 3OH group free, and over here I will be having the 5OH group free. Sorry, 5-phosphate group free. So this strand, at the end, what will I have? 5- phosphate. Over here I will be having 3-OH. If you see, they are opposite. They are anti-parallel. They are not parallel, they are anti-parallel. One is in 3- to 5- direction, other one is in 3- to 5- direction in the opposite way. And what do you call this structure as? You call this structure as a nucleotide. Now if A is there and T is over here, then how many bonds will be over here? To combine both of these strands, you need bonds, right? You have 2 hydrogen bonds between A and T. If it was G and C, you would have had 3 hydrogen bonds. Now this structure of DNA was given by whom? Watson and Crick, the double helical model structure. So this basically forms the double helical model structure over here. Okay, and now one very important thing that I want to tell you along with this is that this is a strand, this is a strand. I'll write this is 5-, this will be 3-, this is 5-, then oh wait, this is okay, just a second. This goes like this, so this is 3- and this will be 5-, right? Now this structure which is formed over here, one whole helix, this you call as one helix, okay? This is one helix, my dear children, and in one helix you will be finding 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. You will be finding 10 base pairs, 10 base pairs as in A and T, G and C. This is a base pair, a pair of bases, nitrogen bases. One base pair, second base pair, third base pair, you will have 10 of them in one helix, okay? And the distance between them, the distance that you see over here, 0.34 nanometer. Okay, it is 0.34 nanometer. Got it? So whole length of one helix will be what? 10 into, so 10 base pairs are there in between them, this is a space, so I'll say 10 into 0.34 will be the whole length of this particular strand. Okay, these are the basic things that you should be knowing. These are the salient features. We will be reading it one by one. So this is what exactly I formed over here. So you saw phosphate group on the 5th carbon attaching to the 3rd carbon over here. Okay. Now, based on the observation of Maurice Wilkins and Rosalind Franklin. So Maurice Wilkins and Rosalind Franklin were what? The people who did X-ray diffraction. Okay. After X-ray diffraction, the image that we got, that was analyzed by next group of scientists, that is James Watson and Francis Crick, and they were the people who gave the DNA double helix model. Now, this is not for you to read as of now, this is for you to read later on, but I am just telling you what should be there in your answer. So, you should be telling that Watson and Crick were the people who gave the DNA double helix model. Okay. According to this, DNA is composed of what? 2 polynucleotide chains. How many chains are there? 2 chains are there, and they are what, children? Anti-parallel. They are each to each other. They are anti-parallel in nature. Okay. The polynucleotide chain on one strand is formed by phosphodiester linkage in 5-to T direction, and the other strand will be having 3-to-5 direction. Okay. Fine. Next thing. Both the polynucleotide chains are held together by means of what? Hydrogen bonds formed between what, children? Nitrogen bases. What are the nitrogen bases? Adenine, thymine, guanine, cytosine. So you have to tell adenine pairs with thymine with how many hydrogen bases? Hydrogen bonds, 2 hydrogen bonds, sorry. And then you have guanine which pairs with cytosine. How many hydrogen bonds? 3 hydrogen bonds. Okay, hence in a DNA molecule, the number of purines, okay, do you agree? The number of purines which will be there will be equal to the number of pyrimidines. Yes, because how many A will be there? That many T will be there. How many G will be there? That many C will also be there. Both the nucleotide chains take a turn for every 34 angstrom. That contains roughly 10 nucleotide base pair, hence the internucleotide is 3.4 angstrom. So in angstrom also you can write the same. Okay, in angstrom also you can, or basically I can just make it over here only. This is one helix. In that they are saying this strand, this distance between two base pairs is 3.4 angstrom. Instead of nanometer, you have written in angstrom. Now if you have 10 of them, total will be what? 34 angstrom. The total length will be 34 angstrom. Okay, works? In DNA molecule, the pentose sugar and phosphate form the backbone of DNA. So this is the sugar that we have, pentose sugar, as in they has 5 carbon, that is why it is known as pentose sugar, and it also has phosphate, that is why we say there are pentose sugar and phosphate forms the backbone of the DNA. Pentose sugar, this is the sugar, and this is the phosphate which is forming the backbone of the DNA molecule. So I hope you would be able to write the salient features or DNA double helix model. Will you be able to write it? Okay, it's easy, no? It's not very difficult. I hope you will be able to write it. Next question that we have is, DNA replication is fast, accurate, energetically expensive. Okay, fast as in it has to occur very fast, accurate as in there should not be any mistakes occurring over there. Okay, energetically expensive as in there should be too much amount of ATP being utilized in this particular process, and substrate and enzyme dependent means that you need something for formation of the DNA and you need enzymes also which would be helping in the whole process. Now it is initiated from a specific site. It should be initiated from a specific site and cannot uncoil on its entire length. Cannot uncoil on its entire length as in, for example, I cannot just open the DNA during the entire length. If I do so, what will happen, children? It will take too much amount of energy, because you know hydrogen bonds are there in between, you have to break all of them. So DNA replication is all about these things that you see over here, but you have to explain the process of DNA replication considering all these features. So assuming that you might be knowing this thing, the person who have asked this question is telling you to write a process of DNA replication which should include all these points. Now this is a model question that came in 2024-2025 paper, so we have to solve it. What do? We just focus on DNA replication method that we know. One step of DNA replication is initiation, okay, then is elongation, and then is termination. As simple as that. In that we have to mention all these things. So how will we mention? I will be telling you for that. So first step is initiation, where basically, suppose, do I have a picture of it? No. Suppose this is the DNA strand, okay, and they are bonded over here. I want to make a replica of it, so basically I want to open it. So I will be opening it one by one, my dear children. What will I do? I will be opening it one by one. First I took this particular strand. So when I opened it like this. Others are still being bonded. Why? Because if I try to open everything, it will take lots and lots of energy. So the first step that I have is initiation. I have to initiate things, and initiation will be happening at one particular site of DNA which you will be calling as ORI site, or origin of replication. Now that is something that you wanted. There is a specific site for initiation. Yes, there is what site, children? Origin of replication, or ORI. So initiation begins at the specific origin of replication. Then what happens? Then the bonds which are over here has to be broken down. For that you need the enzyme. What is that enzyme, my dear children? The first enzyme that comes to play is DNA helicase. It will be breaking the bond. So here you need enzyme. It is enzyme dependent. That was one part of the question. So helicase unwinds DNA at specific location, and this thing, no, this fork like thing that is being formed over here, this is known as replication fork. What do you call it? Replication fork. Okay. Now single strand binding protein stabilize unbound DNA. So these guys can come back over here. So for that you have another proteins also which are known as single strand binding protein. So onto the single strands the proteins will be coming and binding. So that does not allow the recoiling of the, or basically binding of the strands that you see over here. That point, if you don't want to write, don't write. It's okay. It's okay. Next thing. Now you have opened it by DNA helicase. You want to make up a new strand. For that you need another enzyme which is known as DNA polymerase. And DNA polymerase also makes the DNA in 5' through 3' direction. Remember, RNA polymerase was making the RNA in 5' to 3'. Similarly, it will choose a strand that is in opposite polarity. If it has to make 5' to 3', then over here it will choose a strand that has opposite polarity, that will be this. And this strand will be like this. So onto which strand will it go and bind, children? It will go and bind over here. It will go and bind over here because you have to make a complementary pair, right? But the problem is that it cannot, DNA polymerase cannot initiate the process of DNA replication without a set of nucleotide already being there. You already need certain nucleotide over there. So for that, what do you call these set of nucleotides which are already being provided? You call it a primer. So primer is being made first. So primer is a complementary pair that would be made first, children. So primer is over here. I'm not writing it. Okay. And then onto this, the DNA polymerase will come and make this complementary strand. For suppose here you have A, here you have T, then here it will be T, here it will be A, and the thing goes on in this particular direction. More and more it will be opening up, more and more things will be forming. So this particular strand that you see over here, where the DNA replication is occurring very smoothly, which is in 3' to 5' direction. What do you call this strand as? You call this strand as leading strand. What do you call it? You call this as leading strand. Now the problem comes over here. What is the problem? DNA polymerase always makes things from 5' to 3' direction. Agree? 5' to 3' direction. And it always needs what, children? A primer. Always needs a primer. So over here what happens is that every time a primer has to be added over here like this. So it will go up to some certain length and then will stop. Again it will go up to certain length and then will stop. So now the DNA polymerase will come and act and increase the length, increase the length, increase the length. That is how it will be made. So is it continuous or discontinuous? It is not continuous, it is discontinuous. This strand will be forming discontinuously. This strand I call it lagging strand. What will I call it? Lagging strand, because it is lagging actually. Okay, and now basically these primers that were added, it has to be removed and they have to be joined together to form a proper DNA. So these small strands that you see over here, what do you call them? Okazaki fragments. And the Okazaki fragments, basically they have to be rejoined together with the help of another enzyme which is known as DNA ligase. DNA ligase will come and join it. So over here I told enzyme is needed. I already told that yes, it is speedy process that is happening over here, it is accurate. Okay, for accuracy, what will I show? That basically there will be another set of enzyme that will come and check if everything is complementary to another thing. For example, T goes and pairs with G, that is a problem. So it will be rechecked. So all those things happens, my dear children. So that is the funda behind it. So you have to explain everything that is DNA replication in detail. When you explain DNA replication in detail, you are basically including all the points that was asked by the questionnaire. And one very important thing over here, whenever I am saying I need T, whenever I am saying I need A, what are these things? These are the substrate that I need for DNA replication. I call them nucleotides or polynucleotides. There will be so many polynucleotides that I need for the formation. Okay, so let's read it now and it will be more easy for you to understand. So initiation means begins at specific origin of replication. Helicase will be unbinding the DNA. Okay, and then primase synthesizes primers to provide a starting point for DNA polymerase. So another enzyme, that is primase, will be coming and forming the primer, which will later help in the formation of DNA with the help of DNA polymerase. Next thing, when I am elongating the process, this is initiation. I mean elongating the DNA. For that, DNA polymerase synthesizes the new strand in 5' to 3' direction. Leading strand is synthesized continuously. Lagging strand is synthesized in short Okazaki fragments, joined later by what? DNA ligase, which are there. Proofreading by DNA polymerase ensures accuracy. There will be another type of DNA polymerase which will coming and checking and saying that whether everything is fine or not. So I have included all the points. Next, termination. This process has to get over also, right? So occurs when replication fork meet or reach termination. So probably there might be replication happening from that side also, from here also. So these replication fork coming and conniving basically ends the termination. Enzymes remove RNA primer and gaps are filled by DNA polymerases. So there will be enzymes which will be removing these primers later on, which were added, and the gaps will be filled by DNA polymerase. Now the DNA polymerase has a strand, right? It needed the initial support, so it took it. Now it has formed the strand. Now it will take support from these strands and make the whole strand over there. DNA ligase seals the final nicks in the backbone. Whatever gaps are there, it will be sealed by the DNA ligase as well. And that's how you have finished this. So you have to explain DNA replication process. Without explaining, you cannot expect the person to give you marks. Okay? So do not mug this up. Try to sit and understand. When I said you something, I hope that you understood what I tried to tell you. Okay? Fine. I hope you're good. Okay, so next thing, it's an easy question. This will boost up your mind. Now, given below are sequences of nucleotides in particular mRNA and amino acids coded by it. So this is the mRNA. Okay, this is sequence of what? mRNA, and the mRNA, as I told, they will be coding for amino acids, and the amino acid will be coming together to make proteins. Write the properties of genetic code that can be correlated from the above given data. So these are the genetic code. This is a code, 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 this is a code. Okay. So what features can you tell about the genetic code depending on the information that is given to you? So I know one thing, that AUG is basically coding for methionine. UUU is coding for phenylalanine. UUC is also coding for phenylalanine. That means there can be two separate codons or two different type of codons that can be coding for what? One amino acid. So UUU is one, UUC is another one. Okay, now GAG is coding for glutamine, UUA is coding for leucine, GUG is coding for valine. What about UAA? It is not coding for anything. So can I say it's a stop codon? So I understood two things very easily. One is there could be two codons or two or more codons for one amino acid. Okay. Second thing, UAAA is not coding for anything. Basically, it can be a stop codon. Okay, that is all something I understood. One more thing that I understood is basically nowhere things are getting stopped. It is happening continuous manner. So these codes are contiguous. What do you say? It is contiguous, or basically this process is a continuous process. Okay, or I can say genetic code, since I am talking about genetic code, they do not have any full stop or comma in between. They are just continuous in nature. So at least these things I got to know, and I also got to know one more thing. Basically, there are three such nucleotides coming together to form what? A codon. So there are codons, basically nothing but they are in the form of triplets. Okay, and this feature, when I am saying the two or basically more codons are there for one amino acid, I will say that this feature is known as degenerate feature. Degenerate. For example, we suppose that we have our best friend has just one friend, but our best friend has so many best friends. So basically it's degeneracy. So that is degenerate. Okay, that is one particular feature. Then I got to know that its codons are basically contiguous. Then I got to know there is a stop codon as well. And stop codon does not code for any amino acid. Okay. I also got to know one more thing, that is there is AUG, and AUG, what I studied, it was a start codon. It was a start codon. It was helping in starting the process of transcription. But here I see that it is also coding for methionine. So basically, I can say that AUG has dual function. It has two functions. One is to act as a start codon, other is to basically help in the formation of methionine. Okay. So 1, 2, 3, 4, 5 things I could find out. Apart from that, there is also one more point. Let's see. So codon is triplet. Got it. In which 61 codons, total we have how many codons, my dear children? We have 64 codons. 3 of them are stop codons, UAA, tell me, I taught you earlier, UAA and 2 more stop codons you have to tell me, okay? So, apart from these 3, all of them are coding for something. 61 codons are coding for something. Next thing, AUG has dual function, my dear children. It codes for methionine as well as it acts as an initiator codon or a start codon, okay? Some amino acids are coded by more than one codon. So, you call this code as degenerate code. Okay. The codon basically are read in contiguous fashion. It is read in contiguous fashion. There is no full stop, there is no comma. Over here I already told, but still. So, you have UAA, UAG and UGA as terminator codons. That is also one thing that I can find out. Next thing, can I say that this is basically a universal thing? In every other organism, you will be finding that this codon is coding for this amino acid. So that is also one thing, that the codons that you see is nearly universal. For example, from bacteria to human being, UUU would be coding for phenylalanine only. Some exceptions are obviously there. For example, you find it in mitochondrial codons as well as some protozoans. So in protozoans and mitochondria will also have their own codons and things like that. They have their own DNA. So right, so they might have certain changes, but in general, if we say, it is kind of universal. So these are the features. So if you're asked about the salient features of the genetic code, or if you're given a question like this, you have to frame the same answer which is coming like this. So they will try to twist it up and give it to you, but you have to take it as a cheese and eat it. Okay, as easy as that. Works? Next question. Describe the steps involved in DNA fingerprinting technique. The steps is already discussed when I spoke about VNTR. Okay, so isolation of DNA is extracted from the sample. That can be blood, hair and saliva. Okay, in this we can get our DNA. Digestion by restriction enzymes. So there will be restriction enzyme that will cut the DNA. Okay, and then it is done with the help of restriction enzymes. Okay, separation by gel electrophoresis. There will be strands that will be separated along based on their length and everything. Transfer to membrane. So this is known as southern blotting, where I am transferring all the content to a membrane. Now I will be hybridizing with DNA probes. So radioactive or fluorescent probes, complementary to the specific sequences, bind to variable regions of the DNA. If they are binding, they will show up the color change or probably anything. Now we have to visualize it. That is the last step. The hybridized fragments are visualized using X-ray film or UV light, producing a unique DNA pattern that is not same for all the human beings. So this pattern is compared for identity or relationships. Okay, so with DNA fingerprinting, okay, what all can you find, children? Basically, it will be helpful for finding the culprits. Also, if you want to find the parents, okay, we are confused about who's the parent, and there can be a DNA test that can be made, because it will be talking about this only. That will be really unique. Okay, so all these things are important. So DNA fingerprinting has to be answered like this. We already discussed it, which is why I'm not discussing it again. Works? VNTR, this. So that's what I was telling you. Certain questions now are framed in different ways, but the content that they want is the same. So don't fall in that trap. Now this question is also ultra easy. Okay, let me see if you will be able to solve it or not. 22nd question. Construct a complete transcription unit with promoter and terminator on the basis of hypothetical template strand given below. So over here, have this strand. Okay, now I have to form what? A transcription unit. A transcription unit consists of promoter. You remember promoter, terminator. Promoter was a region where your sigma and RNA polymerase was coming together. Terminator was a region where the rho factor is coming for termination of the whole process, right? So how can you create a transcription unit if this is a hypothetical template strand? What do you mean by template strand? First question. So suppose this is a DNA, my dear children. This DNA has to form RNA. So there will be one strand which will be actually helping in the formation of RNA, and this strand is known as template strand. Okay, and the other strand where transcription is not going to take place, you call it as a coding strand. So we usually think coding is the one that will code, but no, it's a template one that is going to code. So this is a DNA. Okay, over here you have the promoter region, over here you have the terminator region. Promoter, how do you write? Basically, this is 5' end of the coding, this will be 3' end of the this. So you always say promoter in reference to the coding strand. Everything is done by this guy, so we thought we should be giving some reference to this guy also. So we say promoter is found on the 5' end of the coding strand. Similarly, terminator is found in the 3' end of the coding strand. And over here, what do you have, children? You have the 5' end. Works? Am I correct? Yes. Now, RNA polymerase, which is the enzyme that helps in the formation of RNA, will be moving from 5' to 3'. So if this is 3', it will be moving from here and forming a new strand. It will be coming and pairing up here and making a new strand, my dear children. Right. Now over here, they have asked you that I am giving you the template strand. Okay, this is the template strand. You make a transcription unit like this and give it to me. This is a transcription unit, but the problem is you won't understand which side is 3' and which side is 5'. For that, I'll tell you something. Always remember the area where the arrow goes, that is the 3' end. And over here, it will be the 5' end. So if I want to write, how will I write, children? I can write it as A, T, G, C, A, T, G, C, A, T, A, C over here. This is the template strand. This is the transcription unit that I am seeing over here. This is the template strand, this is the coding strand, this is the promoter region, this is the terminator region. This is the whole transcription unit that I can see over here. Write the RNA strand transcribed from the above transcription unit along with polarity. Now they are asking you to make a RNA strand. This is DNA. You have to make an RNA strand and along with the polarity also you have to write it. So let's make an RNA strand. Are you excited? Let's make it together, okay? Please remember this is, I'll write it properly, okay? I'll write this side properly. So this was TG and then it was C and then it was A and then it was T and AC. So I have been to terminator. Please you don't go to terminator, okay? Or shall I remove it? Wait. Why to make you confused? This is the terminator region. Fine. Let's make the RNA. So RNA will be formed in this direction only. So phi dash is the polarity, my dear children. So A will be binding with what? In RNA, U. T will be binding with A, G with C, C with G, A with U again, T with A, G with C, C with G, A with U, T with A, right, A with U and C with G. And this is phi dash, this is what? 3 dash. Okay, let's check the answer. UACG and AUG at the end. UACG and ATG. Yeah. Oh, they have made it wrong. It's not T, it is U. It's RNA. This is how you have to make the transcription unit. Okay, please make according to what I made like this. Please include the coding strand as well. Okay, that will be more correct way of making things. I hope you got it. Okay, so in the transcriptional unit, you don't have to make this guy. Please don't make this. You just have to mention promoter, terminator region and these things like that. Works? Let's go on to the next question. Come on. List the salient features of human genome. What are the salient features of human genome? Tell me. I told you about the goals of the Human Genome Project. I hope you remember it. Okay. What was it? To identify all the genes, 20 to 25,000. Then after identifying, what do you need? You need the sequencing of the whole DNA. After that, you have to reserve in database. After that, you have to improve the tools. After that, you have to give it to a lot of people who are finding things. Okay. And then you have to make sure that you are not disturbing any ethical things and stuff like that. Now, you're talking about salient features of human genome, which is a direct thing again. You have to remember a lot of content, which is why I'm here. I will try to make you remember things. So, let's do it, children. I have highlighted all the points. Let's do it. Human genome contains 3,164.7 million base pairs. How many base pairs? 3,164.7 million base pairs. Okay. Now, average gene, if I say, it consists of 300 bases in a gene, that is a strand. For example, this is a DNA, this is a gene that will be coding for a protein. This particular gene, so in average genes I see there are 3,000 base pairs in one particular gene. But there is a largest human gene that is known as dystrophin. It is a protein actually. Dystrophin has 2.4 million bases. It's a gene having 2.4 million bases. So how many base pairs do we human beings have? 316.64.7 million base pairs. Now you're talking about one gene. A gene in general has 300 base pairs, but I saw dystrophin which has what? 2.4 million bases. Okay. Now the total number of genes was basically thought to be 20 to 25,000, but it was not 20 to 25,000. It came to be 30,000, much lower than the previous estimates, that is 80,000 to 1,40,000. So it was estimated that this was there, but it was not actually there. It was only 30,000. Okay, and almost all nucleotide bases are exactly the same in all people. So almost all the nucleotides that you see over here between you and me are kind of really same. Okay, now the functions are unknown for over 50% of the discovered genes. So we discovered 30,000 genes, but 50% of them, that means 15,000 genes, we know their function. Rest 15,000 we are not aware about their function also. Okay, now I am telling you one very important thing, it will crack up your head. Less than 2% of the genome codes for protein. Only out of these many genes, and now basically all the content that we have, only 2% of the genome is going to code for proteins. Rest is all useless. Rest is all what, children? Useless. They are repeated sequence. That is the next point. What are those? They are repeated sequence that makes up the large portion of the human genome. And these repeated sequence of the DNA are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding function, but they are really important for the search of evolution and things like that. For example, I want to see how many of them are there in my body and is it similar to yours. So basically I am helping with the study of evolution. Then comes chromosome number 1. We have how many chromosomes, children? How many chromosomes do we have? If you remember, 23 pairs of chromosome are there. Okay, 46 chromosome are there. Now I am saying chromosome number 1 has genes, that is 2968. Okay, 2968 genes. That is kind of really good. One chromosome having these many genes is kind of great. Now chromosome number Y, which is found in males, it has the fewest number of genes, and that is 231. What is it, children? 231. I know it might be difficult for you to remember the numbers, but slowly we'll take it. Okay. Now scientists have identified about 1.4 million locations where single base DNA differences are there. What do you mean by single base DNA differences? For example, I have ATGC, you have ACGC. So this is single nucleotide pair difference. Over here I find just one difference. So similarly in my body also there might be things like that. In one area I might have ATGC, other area it will be ACGC, things like that. So 1.4 million locations are there where single base differences are there, which are known as single nucleotide polymorphism. Okay, now let's read it. So average genome, children, human genome basically has how many base pairs? 3164.7 million base pairs. No, okay. Now average gene contains how many base pairs? 3000 base pairs. Average gene, when you talk about that, it contains 3000 base pairs, 3000 base pairs or bases. But when you talk about dystrophin, it has 2.4 million bases. How many? 2.4 million bases. Then if you count the total number of genes in human genome, it is 30,000. It is what? 30,000, out of which we just know that 50% has some function for coding proteins. Rest 50% we do not know what are they coding for. And now less than 2% of the whole genome is actually coding for proteins. Rest of the things are just repeated sequences. And these repetitive sequences are repeated so many times, dear children, and they are not coding for anything. Now chromosome number 1 has the most number of genes, that is 2968. 2968. Chromosome number Y has the least, that is 231. Cool. And scientists, they have identified that there are certain bases, certain changes in just one nucleotide, which makes 1.4 million locations are there just like that in our whole DNA. This is the salient feature of the human genome. Okay, so write just the major point somewhere. Whatever I have made in dark, just write it somewhere like that, and you would be able to remember it more better. Now you might be able to remember it, but after some day now you might forget, so which is why I am telling you, write it down like that. Okay, you will get the PDF, please don't worry. Okay. Chalo. Describe the lac operon concept of gene regulation. How can you describe the lac operon concept? Let's begin. Let's begin the battle. It's a very important question. What do you mean by lac operon? So basically, there is a bacteria, this E. coli, Escherichia coli. And E. coli loves lactose. Okay, it's a very good medium for providing energy. Lactose is a really good source for providing what? Energy, my dear children. So basically, what happens is that when E. coli is put in this medium, it takes in the lactose and uses energy. But sometimes when lactose is not there, a lot of things happen, and that is what we are going to discuss today. So basically, I shall use this and I'll try to explain. Okay. So this thing that you see is a gene. Over here, you have I gene and you have Z, Y and A gene. How many genes? I gene, Z, Y and A genes. These genes have their own promoter region, that is this P. Okay. Similarly, the I gene also have its own promoter region, that is P. So when promoter region is there, RNA polymerase will come and bind, do the transcription. That's when whatever thing it is going to code, it will be able to code it. What is it coding, children? It is coding a repressor protein. This particular gene that you see over here, it is coding what? A repressor protein. Now there is another gene over here. As I said, it has a promoter region and then there are a set of genes. And it also has one more region which is known as operator region. What is it? Operator region. So the thing with this repressor protein is that this repressor protein, which is made by this particular gene, goes and binds on the operator. Goes and binds on the operator. Now what is the problem with that? Basically, when RNA polymerase comes to make transcript of this, it is not able to go forward. It will stop there. It says that something is over here which is not allowing the transcription of these things. Now what are these things, children? These are genes which will be coding for enzymes. Remember the first MCQ that we had, first or second, I don't remember. It was what? Z is coding for something, Y is coding for something, and A is coding for something. So let's begin it. Listen to me carefully. It's a very interesting thing. This is A, this is Y, this is Z, this is the operator, this is the promoter region. Okay, this is a whole structural gene. This is what the whole structural gene that I have. This is the promoter. This is the operator. Okay, Z, when it is active, it is going to form an enzyme which is known as beta galactosidase. I am going to explain the concept and tell you how to frame the answer. Okay, not one by one, so you have to be with me to understand the concept. Y is going to code for a protein which is known as permease, and A is going to code for another protein which is known as trans-acetylase. And I will tell you the function of them also. Beta galactosidase is the one that will break down lactose. When lactose is there, it will break down lactose. That is a function of beta galactosidase. Permease is the one that will allow the lactose to get inside the cell. It will make pores over here. That is the role of permease. Trans-acetylase is the one that will be breaking down all the metabolites. Whenever this breakdown happens, there will be so many things being formed. Don't want them, something you want, you will be breaking down that with the help of trans-acetylase. Now, as I said, lactose is an energy source. Okay. Now, in normal condition, what is happening is that just parallel to it, there is a neighbor. There is a neighbor over here. And neighbors, sometimes they are good, sometimes they are not that good, right? So over here, they also have a promoter region, because without promoter region, the RNA polymerase cannot come and bind, no? So you need to have promoter region for each gene. So this is one whole structural gene that we have, which has 3 genes in total. Okay. Now over here, there is another one which is known as regulator gene. This is another gene that is known as regulator gene, denoted by I. It is denoted by I. So this regulator gene, when RNA polymerase binds over here, makes the transcript, it is going to form an mRNA, and later it will be forming a protein which is known as repressor protein. What do you call it? Repressor protein. This repressor protein, whenever it sees that the other guys are active, it goes and binds over there, over this. The repressor protein goes and binds on the operator. Now when RNA polymerase comes for transcription, it finds that, hey, operator is there, how can I transcript things? I cannot transcribe them. So that is a big problem, right? At this point, I cannot transcribe all of them, so these enzymes are not being produced also. But it's not like that. Actually, even though it is an off condition, now it is off, okay? The lac operon is not working. It is switched off, okay? But some amount of these enzymes are still being produced somehow little bit of transcription is happening, which is why the permease will be active and the permease will be opening grounds for things to enter and go out. So this is a condition where it's switched off. Lac operon is switched off. Now what happens? I put this E. coli in a medium that has lactose. Lactose will now enter. Lactose will see what is happening over here. I have to save my friend. So lactose basically goes and binds to the repressor protein. When it goes and binds to the repressor protein, now the repressor protein cannot come and bind over here. So lactose is acting as an inducer. because it is going to induce what? The lac operon. It is going to start the lac operon. Lactose comes and forms friendship with repressor protein, which is why now operator is free and the RNA polymerase can go around and basically transcribe all this thing, making beta galactosidase, permease and transacetylase. Now this poor lactose, which actually did so much help, still it is being broken down by the beta galactosidase which is made. Broken down for energy only, for the E. coli to survive. So lactose is a good guy overall. Now we say it's turned on. The lac operon is basically what? Turned on. Okay. Did you understand this? I hope you understood this. Okay, so there are two sets of genes that are playing a major role over here. Regulatory protein or regulatory gene is very much important. It gives repressor protein. Now once you read it, it will become more clear. So switch off metabolism. So when lactose molecules are absent in the nutrient medium, DNA is switched off for protein synthesis. Okay, the regulatory gene produce a kind of protein called repressor that binds to the operator gene. Agree? These do not allow RNA polymerase enzyme to transcribe the structural genes, hence there is no transcription, translation and the required protein is not synthesized. This is known as switch off mechanism of lac operon. Now switch on mechanism. The lactose molecule added in the nutrient medium acts as an inducer which binds to the repressor to form inducer-repressor complex. Both of them, they bind to form a complex. Now the repressor becomes inactive and releases from the operator gene. This enables RNA polymerase enzyme to transcribe all three structural genes, that is Z, Y and A. All these three mRNA strands undergo translation to produce three polypeptide chains. Later, you are directly forming what? Beta galactosidase, permease and transacetylase. These three enzymes together will be breaking down lactose into what? Glucose and galactose, finally providing the energy source. So this is what you mean by lac operon, and you could be finding so many questions around it. I gave you 2 years, but there are so many more also. Works? Done? Easy? Okay? Next, I hope you would be able to do it. Children, you can motivate me also, okay? Just like how I motivate you, you can also motivate me by telling, ma'am, yes, we are participating. I don't want the good words. Basically, if you participate in this whole thing, I'll be really, really happy. Okay, tell me if you are doing things in the comment section. Tell me that, yes, ma'am, we are doing, we are answering, this is the answer for this. I'll be too happy to read your comments down there. Okay, on the basis of the content that I'm teaching you. Chalo, next. DNA replication is said to be semi-conservative. Why? Why do you think DNA replication is said to be semi-conservative? I already told you there is one parental strand and one new strand. Now describe the experimental proof of Michelson and Stahl to show DNA replication is semi-conservative. Now you know Michelson and Stahl did this experiment. Okay, what was this experiment? They were the people who said that it is DNA is replicating semi-conservatively. So let's see how did they do it. So the thing that they did was, they grew Escherichia coli, E. coli only, in a medium that had NH4Cl. What was it, children? NH4Cl. Here the N had, this nitrogen was N15 isotope. Okay, what was it? N15 isotope. And nitrogen is a part of DNA. So basically whatever DNA molecule they made, everything had what, children? Everything, all the strands, they had N15 thingy. Okay, N15 incorporated into it. So all the DNA of the Escherichia coli which was grown in this particular medium had N15 strands of DNA. Now, if I want to separate the DNA strand, no, I will use cesium chloride. What is it? Cesium chloride, which would help me divide them on the basis of density. So when I put the DNA and nitrite, centrifuge it and things like that, what will happen? This particular strand which has both N15 and N15, it will be somewhere around here. It forms a heavy band over here. It's a heavy isotope. N15 is a heavy isotope. Now what I did was, I basically put this Escherichia coli in a medium that had N14 as an isotope. That is another isotope. Okay, I had grown in a medium that has N14. Now Escherichia coli takes 20 minutes for replication. For it to replicate, it takes 20 minutes for them to replicate. So now the DNA that will be formed, let's see how it might look. So this DNA strand will separate out. This is one strand, this is another strand. One will be having N15, this one also has N15. These are the parental strands. Now a new strand will be forming, and this time which nitrogen will be utilized? N14 version will be utilized. So I will find something like this. This is what? N14. This will also be what? N14, my dear children. Now, Michelson and Stahl, what they did was, they again put it in the cesium chloride medium and tried to find what is the thingy that is happening over here. Now, when they put it, okay, now when they put it, they found out that basically it is not coming and settling over the down region. This DNA strand, when it was centrifuged, it was coming over here in the middle. In the middle region. So, isn't it possible, if both the strands were N15, they would have come down over here. Similarly over here also. But I see a new position over here. That means some hybrid might have found. What do you mean by hybrid? Hybrid is something that has new combination. So, over here I see a new combination. So, that is a hybrid, which is in the intermediate level. Which is in the intermediate level. So, I got to know one very important thing, that basically during DNA replication I get one parental strand and one new strand is being formed. So, can I say it's semi-conservative? Yes, I can say it. But let's not stop here. Let's do one more round of replication. Let's do one more round of replication. Another 20 minutes. So, total how many? 40 minutes are done. After 40 minutes, if I still put it in the same medium, N14 medium only, and try to check it, what will I find? So, this two strands I have to separate. When I separate this, I get one strand like this and I get one strand like this. Similarly, this also one strand like this and then one strand like this. And I am putting in which medium? N14 medium only. So, now the new strands which will be having will be like this. Here it will be like this, here it will be like this, here it will be like this. The white one is the N15 one. The white one is the N15 one. Rest everything that you see is N14. This is N14, this both the strands are N14, this is N14, here also both the strands are N14, N14, here one strand is N14. So, now when Michelson and Stahl tried to centrifuge it, they found something like this. Okay, listen to me very carefully. So, they didn't find heavy band again because there was no one like this. They didn't find heavy band. Instead, they found that there was intermediate band and there was light band, my children. So, this is a light isotope, which is why he found light band. Again, then he was sure that it is what? Semi-conservative mode of replication that is happening over here. Otherwise, this would not have happened. Otherwise, you won't be getting two strands like this. One strand might have come from here, other strand might be already there, which is why you get it. So, he got three things out of this thing when he did replication twice. Okay, first he found heavy band, then he found intermediate band, which actually showed that there was hybrid thing happening, which actually proved that basically yes, DNA is replicating semi-conservatively, and over here he found hybrid as well as light band. Okay, after 40 minutes. So, this is the whole thing. This is the setup. Okay, so now how I have to explain? You have to write it like this. So, in both the daughter DNA molecules, one strand is parental and another is newly synthesized, hence DNA replication is known as semi-conservative method of replication. And then he explained the whole thing. Okay, you can go and just have a read of it when you have time. When you will get the PDF. Okay, for getting the PDF, what you have to do? You have to fill in the form that is given in the link. When you fill in the form, you will get the PDF to your mobile phone. It will be given in your WhatsApp only. You don't have to go anywhere. Okay, it will be sent to your WhatsApp when you fill in the form. Please put the correct phone number as well. Works? Okay. Don't worry, we won't be disturbing you with any other calls. So you can join it. Now the next question. I hope that was also clear. Guys, when everything is clear, tell me, ma'am, it's clear. Okay, if it's not clear, you can tell me that also. Now, Oswald Avery and others have continued Griffith's transforming principle to prove DNA as genetic material. Substantiate. So, Oswald Avery and others continued with Griffith's experiment. What do you mean by Griffith's experiment? So, if you are not aware about Griffith's experiment, you won't be able to understand this. So, we have to understand Griffith's experiment, which is very interesting. Pro level interesting. Listen to me very carefully, okay? So, in Griffith's experiment, no, he took Sweetie, the mice. Okay, fine, and he took a bacteria whose name was Streptococcus pneumoniae. It causes pneumonia. Okay, it was what? Streptococcus pneumoniae. This is the bacteria. Now, it has two different type of strains. One is R strain and the other one is S strain. Okay, these are two strains of a same bacteria, R strain and S strain. How it is different, children? R strain 1 has a rough outer surface, whereas S strain 1, no, it has a smooth surface. So R for rough and S for smooth. So S is smooth because it has a smooth polysaccharide thingy that is formed over here, which is why it has a smooth nature. Now, things that appear good are not always good. Okay, this is a life lesson. So basically, S strain was actually the one that causes pneumonia and could kill any organism. So S strain, I will write, it is virulent form of the bacteria. Okay, because it is going to cause the trouble. Whereas R strain is the non-virulent form of the bacteria. Okay, so this was already known. So now let's do the experiment. So first thing that was done was, R strain was put in the mice. R strain. When R strain was put, no, nothing was there. The mice was all normal. So I'll say it was alive. Okay. Then what was done is, the S strain was taken. When S strain was put in the mice basically, it died. So mice died. Okay. That was a problem. So it got to understand that S strain was basically doing something, which is why the mice is dying. Then he was like, I should be doing something else. I should be putting heat killed S strain. That means I have treated this particular S strain with heat. So I am writing heat killed S strain. So when heat killed S strain was put, no, it was S strain, okay, not R strain. R strain was just used once. Heat killed S strain was put in another mice. Okay, I am not making so many mice over here, just understand. Okay, when I am putting it another mice, what was happening? That the mice was alive. When I am putting heat killed S strain, the mice was alive. So I was like, hey, the S strain was killing it. Now when I heat killed it, it is not killing the bacteria. Sorry, it not killing the mice. How come? Let me do one more thing. So Griffith did one more thing. He put a combination of heat killed S strain and a normal R strain, which was not causing any trouble until now. When he put it, he found that the mice was basically again dying. Now Griffith was like, there is something in this particular S strain which is getting transferred to the R strain and transforming it and transforming the R strain. There is something over here which is helping it get transformed to what? A S strain, which is why it is able to kill it. So this was transforming principle experiment by Griffith. But Griffith was not able to explain what was it. He was under confusion. He was not able to find what was it. Now comes what? The role of other scientists, that is Oswald Avery, MacLeod and McCarthy. What they did was, they treated the S strain, heat killed S strain 1, okay. They treated the heat killed S strain bacteria with 3 sets of enzymes, okay. Heat killed S strain first with protease, okay. So in the heat killed S strain and the live R strain thingy, what they added was protease. When protease was added, okay, in this particular thingy, still this particular guy was dying. It was getting transformed and this guy was dying. So I am writing transformation has taken place. Then next thing, because protease are the ones that breaks down proteins, children. That breaks down proteins. So basically it's not the proteins. Even if proteins are getting broken down, okay, or getting damaged, still it is able to transform. The heat killed S strain is able to transform the R strain, and it is why the mice is dying. Next thing, heat killed S strain plus RNase was put. You know RNase, what they do? They break down RNA. They break down RNA. Still, the mice was dying. So that means transformation has still taken place. Next thing, third set of enzyme was DNase. This time when they put DNase, it was damaging the DNA. When this happened, the transformation was not taking place. There was no transformation. That is, this particular mice was not dying, it lived when you put DNase. So probably, what did they conclude from this experiment? That when I am damaging the DNA, that's when it's having no effect. Sorry, it's basically not able to transform. This whole cycle is not happening over here when I am killing the DNA. So probably the heat killed S strain is transferring its DNA into the R strain, which is why it is getting transformed. And now when I am damaging the DNA, this transformation is not taking place. That's when it was understood that DNA is the genetic material that is being passed on generation after generation. Okay, and this was done by Avery and MacLeod and McCarthy. Got it? So this was their experiment. So you have to tell that work to determine the biochemical nature of what? The transforming principle of Griffith's experiment. So he said that there is something that is getting transferred. He called it transforming principle. So they wanted to find out the biochemical structure or the nature of this transforming principle, and they did it by doing this. Conclusion is that protein of heat killed S strain is not the genetic material, because anyway transformation was happening. RNA is also not the genetic material. It was DNA. That was the genetic material. Got it? This is the 26th question, my dear champions. Next thing. Explain the process of translation in protein synthesis. This is the second last question. Explain the process of translation in protein synthesis. How does translation take place? So listen to me very carefully. So we have mRNA. We have mRNA, my dear children, and what do you call mRNA? As messenger RNA. Okay, it carries the message for what protein has to be made in the form of what? Codons. So they have codons AUG, CCA and things like that. At the end they will have a terminator codon, that is UAA or whatever. This is in which direction? 5' to 3'. Okay, and I told you that basically from the nucleus the mRNA comes out to meet what, children? The next organelle, which is ribosome. So ribosome has two subunits. One is smaller subunit, another one is larger subunit. Okay, so mRNA goes and meets the smaller subunit first. So mRNA comes and meets the smaller subunit, which is right over here. Okay, and I'll make it something like this. Instead of making it like this, I'll say this is this is the smaller subunit, and this is the mRNA that we have. So we have A, UG, then CCA and whatever, then UAA, and this is in 5' to 3' direction, and this is what? The smaller subunit. Now, to read this mRNA, there has to be someone that has to come. That is what? A tRNA. That is what? tRNA. So at the background, something is happening with the tRNA also. Let's see what is that something. So this is suppose the tRNA. So tRNA looks somewhat like this. This is the 5' end of tRNA, this is the 3' end, and this thing that you see over here, this is the anti-codon loop. That means it will contain the opposite codon of this present over here. So this is codon loop, this is the codon, and this is anticodon. So over here you will be having anticodon files. AUG, that means it will be U, A and C. This is the anticodon. Every tRNA will be having an anticodon loop, and associately whatever is there over here, depending on the codon which is there, you will be having an amino acid over here on the 3' end as well. Now this process is known as charging of tRNA, or you call it amino acylation of tRNA. So this is happening in the cytoplasm itself. So in the cytoplasm you have the ribosomes, in the cytoplasm you have the tRNA, which is carrying particular amino acids. So over here the amino acid which is coded by AUG is what, children? Did I actually do that? Oh my god. Okay, just give me a second. So I'm so sorry, this is 5, this is 3' end, and here if I say it is UAC, right? Because over here what did you have? You had AUG, and then here you had UAA, here you had 5' end, here you had 3' end, and here you had your small subunit, and this is the anticodon loop, right? Oh my god, this time don't worry, I didn't do anything. So basically depending on AUG which is there over here, which is the codon, we get an amino acid over here as well. So UAC stands for what? Methionine. So basically methionine. So over here, this particular tRNA which have undergone charging, it will slowly come, okay, will come over here and bind with this. So it will appear something like this, 5' and then you have methionine over here, and then AUG, instead of that you have UAC over here. Okay, now what will happen, children? Next thing, the larger subunit will be coming and binding over here. This is what? The larger subunit. Once it has come and bound to AUG, this is the initiator codon or the start codon. Now the process of translation will be starting. How? There will be another tRNA that will be coming around over here, depending on the next codon that we have. There will be another amino acid also. Okay, what will happen is that this bond is broken over here, this bond is broken over here, and over here these guys are attached. Now you will be having another amino acid will be coming. Okay, this ribosome will be moving. Another tRNA will be coming. It will pass on the bond to the next guy, and the same things goes on. It will be whole strand of amino acid that will be formed over here until you reach at the end. At the end, there's a terminator codon. No one be able to, there is no tRNA that will come and bind over here. Hence, termination will be happening. And for termination, there will be certain other factors which will be coming and attaching over here, which you call as release factors. So there will be certain release factors which will be coming and attaching over here instead of tRNA coming and attaching over there. And then it will be finally released. All the amino acids will be forming a protein later on after the processing thing happens. Okay, so this is exactly what is shown over here. You see, a chain of amino acids is being formed. And this is how you have to write the answer. So let's read it. What is translation? Translation is the process of synthesizing a polypeptide from an mRNA template. The process of translation takes place in the cytoplasm of the cell where ribosome is located. Ribosome, what do you call it? You call them as protein factories. Okay. Now activation of charging of tRNA, that is happening simultaneously, I told you. So amino acylation of tRNA is the process of attaching an amino acid to its corresponding tRNA. This is known as charging of tRNA. I want to tell you one very important thing. The amino acids are coming on the basis of the codon, not on the basis of anticodon. It's on the codon. Okay. Now, initiation. mRNA binds to the small ribosomal subunit. You saw that. The start codon is recognized by initiated tRNA carrying methionine. Okay. The one that carries methionine comes first. Then, the large ribosomal subunit assembles forming the initiation complex. Now, the whole process is initiated. Next thing, elongation has to happen. tRNA brings amino acid to the ribosome, matching codons on mRNA through its anticodon loop. Peptide bond formed between the adjacent amino acids. So, what is the bond that is being formed over here? Peptide bond is formed over here. Okay. Now, between adjacent amino acid. Now, what? Ribosome moves along the mRNA in 5' to 3' direction. So, ribosome will be moving throughout the mRNA in this particular direction. Okay. Now, termination has to happen. When a stop codon, that is UAA, UAG, UGA is reached, translation stop as the release factors binds to the stop codon. So, release factors will be coming and binding on it, freeing the newly synthesized polypeptide. So, this I call it as a polypeptide, not a protein. It has to still undergo certain processing. Okay. Works? You can show this in the form of diagram also along with the writing format. Cool, if you have time for that. Works? Okay. Chalo. Next thing. Describe the Hershey and the Chase experiment, which is the bacteriophage experiment I was talking about. Okay. Hershey and Chase experiment was the final experiment to say that DNA is the genetic material. Even though Avery said, okay, Oswald Avery said that this is true, DNA is the genetic material, I just showed you. But still there were people who were finding it difficult to digest that DNA is the genetic material. So now comes the role of Hershey and Chase. So Hershey and Chase, what they did was, they took bacteriophage. They took bacteriophage two of them. Okay. Now, they had it has its own genetic material. Basically, sulfur 35 and phosphate 32. Basically what they did was, where is it? He grew two phages in two separate medium. Okay, in two separate medium it was grown. One phage was grown in sulfur 35 medium, another one was grown in phosphate 32 medium. Okay, so how do I remember 35 and S? Basically sulfur, S for sulfur, and the phi also looks like S. That is how I remember things. So I'll tell you one thing. Over here in the case of a phage, a virus, what is happening is that they have a capsid, and this capsid is made up of a protein, and protein has sulfur in it, which is why they take the sulfur is going to the protein side of it. It goes to the protein capsid. Okay, over here when you talk about phosphate, it is grown in phosphate medium, P32 medium, right? So this phosphate is going and getting incorporated into the DNA of the phage. So phosphate is going to the DNA, whereas sulfur is going to the capsid. Now they are allowed to infect Escherichia coli. They are allowed to infect E. coli. Now when E. coli is affected, no, what happens is that this guy goes on the top of it, basically releases the genetic material inside. It releases genetic material inside. The rest of the things, basically this particular capsid that is being there, which is coded by this. So I will make it in yellow. Okay, this stays outside. This stays outside. Thus the genetic material is getting inside. Over here, as I said, this guy will also go and affect. Now, its genetic material is inside. What about the capsid? Capsid is outside only. It will look like this. Okay. Now what was done? Basically centrifugation was done. Okay. Basically centrifugation and everything was done, and it was found that in the case of this, the bacteriophage which was grown in S35 medium, no, in that the supernatant, or basically whatever you find at the the fluid, the liquid solution which is over there. Okay, just a second, I'll just change the color. It had S35. What about the Escherichia coli? The Escherichia coli, this area, no, you call it as pellet. Sorry, this area you call it as supernatant. Okay, supernatant, which is the fluid thingy that you find when you centrifuge things, no, at high speed. Okay, everything that is denser settles down, and everything that is lighter settles up. So liquid region settles over here. Okay, and the pellet region, it stays over here. So it was found that in the pellet there was no S35. There was no S35. So S35 was found in the protein only. S35, we know it is the protein. So basically it's not the protein that is getting transferred into the organism which it is affecting. It is not the protein that is getting transferred into the organism that it is affecting. On the other hand, when I saw this over here, I found that Escherichia coli, basically they were affected, and in the pellet I was able to find what, children? P32. That means it was the DNA that was getting moved to the Escherichia coli and affecting it. In the supernatant, I found nothing. With this experiment, they got a solid proof to say that it's basically the DNA which is moving from an organism to the other organism. Here, bacteriophage is transferring its genetic material, and this genetic material is the DNA, not the protein, because it was not found, the protein was not found in the pellet, it was found in the supernatant. But over here in the pellet, what did you find, children? P32, which was phosphate, and phosphate is going and attaching to the DNA. It is helping in the formation of DNA. So DNA was the genetic material. Got it? Now we will have a quick read of it as well. So grew phages in two separate mediums, one with radioactive S35 to label the protein coat, okay, another with radioactive P32 to label the DNA. Infection. First stage, radioactively labeled phages were allowed to infect E. coli bacteria. Then what was done? Blending. The mixture was agitated in a blender to separate the viral protein coats from the bacterial cells, because the bacterial cells are there and on the top of it the viral protein coats are also there. So to separate it, you agitated it with the help of a blender. Then you centrifuge things. The solution was centrifuged. In the pellet it contained the bacterial cells, and the supernatant it was containing the viral protein coats. Okay, the supernatant it was the fluid along with the viral protein coat. In the pellet always it was the cells. Okay. Now what his observation that was found was, in S35 labeled phages, radioactivity was found in the supernatant over here. That means it is found, the protein is still there, it has not got in, the capsid and everything is there only. Whereas in P32 labeled phages, radioactivity was found in the pellet, that is DNA entered the DNA basically it entered the bacteria, which is why you could find it there. So this was the experiment done by Hershey and Chase, and this was widely accepted. This was said that yes, it is basically the genetic material, that is the DNA is the genetic material and nothing else. So first done by Griffith, then by Avery and MacLeod, and then by Hershey and Chase. And with this, I know it's 2 hours, I don't want to go any bit ahead. I just want to say thank you. I hope you understood each and everything. I hope that I was helpful. This chapter is lengthy, which is why I had to take it forward to explain you a lot of things. But I think I try to explain you in a way that you understand. Okay? So I wish that you understand. I hope for that and I pray for that. And I also wish you all the very best. Definitely do not forget to subscribe to our channel if you think that this videos or this series is useful for you. And definitely do not forget to let us know your love as well. Okay, and one more very important thing, if you want the PDF, the link will be given in the description box. Fill the form, you will get the PDF. Fine. So see you on Monday. On Monday, Chemistry Ma'am will be starting off her next chapter. Cool? Works? So bye-bye. Take care.